Okay, we will go ahead and get started. Good morning, everybody. Good afternoon, good evening. Welcome to May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. This is our second day. We worked a whole year to prepare for this event. We have a wonderful group of speakers and many participants signed up for this event. Unfortunately, it is not possible to do this event in person. However, we are very happy that we have the opportunity to include more participants and more of our colleagues and friends in this online format. So most importantly, I hope you are all healthy and safe and can manage these difficult times as well as it can be. And we also hope that this program and the online form will be useful to you and it will allow you to make the best use of this time. So we have 2 weeks of events planned. And so today we have a very exciting presentation, series of presentations by Lindsay Pino and Brian Shorrell that focus on targeted analysis with Skyline PRM and also introduction to data independent acquisition. So let me first introduce our speaker, the first speaker, Lindsay Pino. So Lindsay is a postdoctoral researcher of Garcia in the University of Pennsylvania, where she now uses quantitative mass spectrometry to study genetics. Before that, Lindsay earned her PhD from the University of Washington Genome Sciences, working under the direction of Mike McCoss and Bill Noble. And so during this time, she developed techniques for data independent acquisition and did a lot of work in both developing new methods, but also making educational resources available in this field. And before joining Mike McCoss's lab, she spent 3 years working at the Broad Institute in the lab of Dr. Steve Carr, where she collaborated with Sue Abatiello on developing targeted SRM experiments. And very recently, I learned that before all of that, Lindsay spent 2 years in South Korea as a Fulbright scholar. And so she has already quite a bit of experience in different types of research in different parts of the world. So Lindsay, we are very grateful that you were contributing your time and your effort to this program, and I very much look forward to your presentation. Thank you so much.
Thanks, Olga, and thanks for everyone who's joining in. We've got almost 263 people that have already joined. So I'm going to do my best throughout this presentation to try to catch any of your questions. And if I, for some reason, miss one of your questions, feel free to reach out to me on Twitter or through email. All my contact information can be found in the documents Google Drive on this course.
Oh, actually, I forgot to mention, let me do this indeed.
Yeah.
So for the participants in terms of logistics, so we have 2 functionalities. One is chat and the other is Q&A. So you can see it at the bottom of your screen. So in the chat, you can post, comments and suggestions. You can post them to the participants, to the panelists and attendees, or to panelists only. But also we have Q&A button. And if you don't mind, please use Q&A to ask questions, because it gives us a functionality to queue the questions and to kind of handle them a little bit better. And so Lindsay will be able to read your questions from the Q&A, and if she doesn't, we will read them for everyone, so that we will make sure that your questions are answered. And Lindsay told us that she really doesn't mind to be interrupted, so don't wait until the end. Please ask questions as it goes, because it's important to understand the beginning of the presentation well enough, right, to benefit from the end. So
always.
Yes.
Okay. So the way this first workshop is going to be structured, I'm going to give a little bit of background in targeted mass spectrometry. This will be a lecture format for this first half of the workshops today. And then after this short lecture, we're going to be getting hands-on with Skyline. Overall, what I'm hoping all of the attendees are going to be able to come away with is the ability, one, to assess the experimental pros and cons of using targeted proteomics in comparison to something like discovery proteomics. In this workshop today, we're going to be focusing on targeted and quantitative proteomics. For the next thing, for our more biomedical researcher colleagues that might be joining to learn more about mass spectrometry and proteomics and how it might help your research, I'm going to explain a little bit the fundamentals of mass spectrometry proteomics, including things like peptide fragmentation and just the real basic components of a mass spectrometer. We're going to walk through the different steps of a targeted proteomics workflow and the information required to build especially a parallel reaction monitoring PRM assay. And overall, throughout this talk, I'm going to be interspersing these slides pointing out some of the functionality of the Skyline software suite, which can be used throughout the entire experimental workflow for quantitative mass spectrometry proteomics. So the format in this lecture half, I'm going to use this very bold and obvious slide format when I'm pointing out features of Skyline. So it's going to have this blue Skyline background. It's going to have screenshots of Skyline. And it's going to kind of be pointing out these different things. So one way you might want to attend this lecture is maybe you want to have Skyline open in the background to kind of move back and forth when I put up these kind of orientation slides for the Skyline ecosystem. And this way you can kind of see for yourself hands-on what some of these different buttons are doing and what happens if you press them. So first off, we're going to start with some of these fundamentals of mass spectrometry proteomics. So much like all other types of research we're doing right now, mass spectrometry proteomics really have to balance three experimental aspects: the research progress in your daily life, your sleep, and your work-life divide right now might be three experimental aspects of day-to-day life that you're trying to balance. Mass spectrometry is much the same. The three things we're trying to balance in mass spectrometry proteomics is the scalability of an experiment. So how many samples you might be able to run if it's high throughput or low throughput. Whether or not the peptide detection is comprehensive. And by this, we mean, are you measuring hundreds of things or are you measuring thousands of things in a single mass spectrometry experiment? And then finally, how sensitive is the quantification of your peptides that you're measuring? So these are really how the two approaches to mass spectrometry end up shaking out. On one hand, on the right-hand side here is discovery, sometimes also called shotgun proteomics, mass spectrometry proteomics. So these methods, these mass spectrometry methods, are scalable in that you can run lots and lots of samples. They're very comprehensive. You get a great overview of all the different peptides or proteins that might be in a sample. But what you're sacrificing is how sensitive the quantification is if you're doing discovery or shotgun proteomics by data-dependent acquisition, DDA. On the other hand, on the left is targeted, also known as quantitative proteomics. And in these methods, they're also very scalable. You can run lots of samples, and you also get very sensitive quantification. What you're sacrificing is, at least in the past, how many of those peptides you're able to measure. So in this talk, we're going to be focusing mostly on parallel reaction monitoring and how to build a targeted or quantitative proteomics method for parallel reaction monitoring mass spectrometry. But coming up next after this talk, Brian Searle is going to be talking about data independent acquisition, sometimes also called SWATH. And this is kind of the latest, greatest emerging technology in mass spectrometry that everyone in the community is kind of excited about because it promises to bring all three of these things together. So both mass spectrometry approaches, whether you're doing discovery-based or whether you're doing a quantitative experiment, all begin with a kind of common sample preparation. The first is you have whatever your experimental system is, if this is animal or tissue-based or cell culture. Getting the proteins out of the samples can require a lot of various sample manipulations, including tissue homogenization, cellular lysis. If you're interested in specific subcellular fractions, you might isolate mitochondria or nuclei. Once you get the proteins extracted out of your sample, you might do other forms of sample manipulation. For example, you might fractionate all of the proteins from your sample. You might enrich for certain post-translational modifications. And something that is becoming more and more common is doing immunoprecipitation to enrich for one protein or one set of protein interactions. For bottom-up mass spectrometry proteomics, the next step after you get the proteins is to digest them, enzymatic digestion. There's a lot of different options for this, and it kind of depends on your experimental needs, but the most common for sure in the community is using trypsin to cleave at lysines and arginines. We'll be talking a little bit more about this and in the context of Skyline in just a bit. And now finally, you get peptides for mass spectrometry. Sometimes you can do additional enrichments at the peptide step. So you might do a second round of enrichment for post-translational modifications like phosphorylation. Very commonly, we perform a sample desalting to remove all the reagents that were added to the sample upstream of these peptides. And at this point, now you have some mixture of peptides that is ready to put on the mass spectrometer. Doesn't matter at this point if you were preparing to do a data-dependent acquisition, a shotgun acquisition, or if you were going to do a targeted mass spectrometry experiment. You have these peptides for mass spectrometry. Most mass spectrometry workflows are hyphenated with liquid chromatography to simplify the sample. So this is a part of mass spectrometry that sometimes in the community we don't talk about enough, but we're all highly dependent on this liquid chromatography step. The peptides are ionized and introduced to the mass spectrometer. For the parallel reaction monitoring that we're going to talk about today, we're going to focus on how to select precursors. So this is the peptide sequence plus the charge. Is it +2 ion? Is it a +3 ion? And then we measure all of the fragments for this precursor. And what we get in the end in a quantitative mass spectrometry experiment is something called a chromatogram. So we're going to go into more depth on a couple of these different steps. But just to show you kind of the overview, the big picture forest, so to say, Skyline supports each one of the steps in this experiment. So when we start an experiment with a hypothesis, Skyline can be used to help build which proteins of interest you might want to develop your assay for. During assay development, Skyline has several features that are going to help you optimize this method, including things like retention time calculation. For DIA later, Brian is going to touch on a couple of the features that Skyline is going to be able to help you with. Whether you're using a triple quadrupole style mass spectrometer, a linear ion trap, an Orbitrap, a TOF instrument, Skyline can process all of these different types of data from whatever vendor you happen to be using in your lab or with the core that you're getting your samples run with. Skyline will process all of the data. There's core analyses within Skyline that we're going to be walking through in just a bit. And then there's even additional analyses that you can add on to your Skyline software. These are called external tools. And I invite you to go to the Skyline website, look at the external tools store. They're all free. And they're just additional pieces of software that you can add on to your Skyline suite to help you perform things like biological inference and visualizations and statistical quantification. For example, MS Stats is an external tool that you can add on to Skyline. So let's talk a little bit about my dog and how cute she is. So hypothesis generating research versus hypothesis driven research is really what divides whether you're going to do a discovery based proteomics experiment or whether you're going to do a targeted or quantitative proteomics experiment. So if we look at this picture of my dog, there's a couple things about this picture that we might be seeing and not thinking too much about. For example, In this picture, there's a couple different features. There's the mountains in the background. There's obviously this dog in the foreground. We know she's in the water. Looks like she's at the beach. Overall, when we look at this picture, the fine details are not super important. Like how calm the water is not very important. The exact layout of the beach is not important. Really, the main focus of the picture, the dog, is extremely easy to pick out of the picture. So if we were to think about this as a mass spectrometry experiment, if the ground truth, the biology that we're trying to study is on the left here, if we do a discovery or shotgun-based proteomics experiment, we'll lose some of the fine detail. So we might not really be able to tell the waves. We might lose a little bit of the mountain in the background. We might not see so much detail on the beach, but we can still pretty much tell this is a dog in a general outdoor environment. With targeted, however, we can zoom way in on the focus of the picture and see this is a dog wearing a bandana. Maybe she's in the ocean. It's kind of hard to tell. Where targeted proteomics really starts to shine, however, is when measuring everything isn't useful. So here's another picture of the same dog in a different context, a different matrix, you might say. The main focus in this picture is a lot less focused and more difficult to see because we have all this noisy background that we don't actually care about. So the most abundant feature in this picture is this grass, this long grass, but we don't really care about it. So now if we were to do a discovery-based proteomics experiment on a sample such as this second picture, we lose what we're actually interested in because our mass spectrometer is focused on sampling all of the things we're not interested in. So depending on your experiment, it makes more sense to do discovery or more sense to do targeted. We're focusing on experiments where you know exactly what you're interested in. You're interested in the dog in the picture, and you really want to focus in on getting as much detail, as much quantitative information about those things that you know you're interested in. So I'm going to pause real quick and see if there's any questions so far, kind of comparing the sample preparation workflow that's shared between discovery and targeted. And it looks like we're doing okay for now. So I'm going to move on to this next part of the lecture. So bottom-up proteomics, like I had mentioned, begins by preparing peptides from the proteins in your sample. So here I'm just representing a transmembrane protein that has, 1, 2, 3, 4, 5, 6, 7, 8 different transmembrane domains. Each one of these circles represents a different amino acid. I've colored in red amino acids that are lysine or arginine. So we're going to extract the protein from its biological context. So this was originally a transmembrane protein. We extract it out of all the membranes and we linearize it. So already we might want to be thinking about, we just lost the context of where this protein exists in the cell. We've linearized it, so now we no longer have the structural information of this protein. And now we've digested it. So now we actually have peptides. We don't know the original protein necessarily that these peptides came from. The next step in the workflow is to separate all of the peptides from our hypothetical protein by liquid chromatography before it enters the mass spectrometer. So these peptides are separated by their physiochemical properties, so namely their hydrophobicity. The retention time of the peptide indicates the point at which the peptide elutes off of the liquid chromatography column. So typically, very short peptides elute first, and typically, very long peptides take longer to come off of the column. So what this means is that in the mass spectrometer, as we're making measurements, and the two measurements we're making in the mass spectrometer are the mass to charge and the intensity of these ions. Typically at the beginning of a liquid chromatography gradient, we're getting these very small hydrophilic peptides. And then at the end of the gradient, we typically get longer hydrophobic peptides. And we're measuring their mass to charge. We're measuring their intensity as they go into the mass spectrometer. So these peptides, once they're in the mass spectrometer, are going to be broken into fragments. So there's a couple terms here that if you're just joining our mass spectrometry community, we are unfortunately a very jargon-filled community. So the peptide itself, composed of the amino acids, makes up the peptide sequence. The peptide sequence, based on its amino acids, has a particular mass. But a mass spectrometer measures ions, so it also has a charge. So for example, this peptide sequence, ADT, blah, 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 R, has a mass of 1,417 and a charge of 2. What we actually measure in the mass spectrometer is the precursor or parent ion of this peptide sequence, which here is denoted by the peptide sequence in amino acids, plus the charge, which for this peptide is +2. So we measure this precursor in the mass spectrometer. We measure its mass to charge, or MZ, which is typically written as this numerical format, and sometimes we will add these plus symbols on the end to signify that it is a +2. When this peptide, this precursor, enters the mass spectrometer, the next thing we do in the mass spectrometer is produce fragment or child ions. This happens over the course of a whole community of precursor ions. So one particular precursor ion might fragment at this first amino acid A, producing a very short fragment and then a very long fragment. We might fragment on the opposite side of the precursor. We might fragment in the middle of the precursor. And over the full population of all the precursor ions, we get different population of different fragments. We signify these fragments and where the fragmentation event happened with this fragment ion notation, where we start with B's and Y's in this lecture, because this is most commonly what we see in typical conventional bottom-up mass spectrometry. But there are lots of other different ion species that can be generated by mass spectrometry. Typically, each precursor will only break once, will only fragment once in the peptide backbone, but you get a whole population of different breaks. Some breaks are more common than others, and that contributes to the intensity of a particular fragment ion. In a perfect world, all of the fragments would happen equally with equal probability, and you would get a mass to charge spectrum like I'm showing on the bottom left. So on the horizontal axis is the different MZ values for the different fragments that I've also noted in the picture above, the cartoon of our peptide, and then the relative abundance. So in a perfect world, every single one of these fragments would be just as likely as any other fragment. However, that's not the case. Typically, when we see mass spectrometry fragmentation patterns, some fragments are much more likely and much more intense than others. Typically, depending on how you set up your mass spectrometer, you can change the distribution of these fragments. But in a standard experiment, you would see some fragments more than other fragments, and that would show up as their abundance, their relative abundance being more or less. If we were to stack multiple mass spectrum on the right as a function of time that they're eluting off of the liquid chromatography column, we get what's called a chromatogram. We're going to go over this again in just one slide. So each one of these individual points on the chromatogram as we march up the curve and then back down the curve represents another mass to spectrum scan in the mass spectrometer. So I'm going to illustrate what I mean by that. So I'm going to show the chromatogram on the left, and the circle is going to march up the chromatogram and then down the chromatogram. At the same time, I'm going to be showing each one of the spectra, the mass to charge spectra. And then finally, I'm going to have different points marching along the retention time. So we start at, let's say, minute 44 in the chromatogram, and we see almost no fragments in the mass to charge spectra. Therefore, the intensity in the chromatogram on the left is 0. As we move down retention time, we start to see the fragments of our precursor increasing because more precursor, more peptide is eluting off of the liquid chromatography column. We see more intensity in these 3 fragments marked in red. So we see the intensity in the chromatogram start to increase. As we move farther in the elution profile of the peptide, we see in its spectra, the fragment ions are becoming more intense, which we see reflected in the chromatogram. At the height of the elution of this peptide off of the column. The fragments are their most intense because we have the most peptide going into the mass spectrometer, and we see the apex of the chromatogram on the left. Now the peptide is finishing up its elution off of the column. We've eluted almost all of the peptide off the column, and therefore its fragments are starting to get less intense until finally the peptide is done eluting off the column. So we've stacked all of these spectra into a chromatogram. The chromatogram is what we use in quantitative mass spectrometry to measure whether one peptide in one sample is more or less abundant than a peptide in a different sample. So we use these chromatograms as the quantitative measure of how much precursor, and therefore we assume how much peptide, and therefore we assume how much protein was in the original sample. We view chromatograms in Skyline. Just like I was showing you, all those chromatograms were generated with Skyline previously. I'll point out a couple things in Skyline, and then I'm going to take some questions. So first, we talked about protein, peptide, precursor, and transitions. Skyline will show you all of these in these nested trees that I've pointed out on the left that are in the target window. Proteins break down into their peptide sequences. Peptides can have more than one precursor. Here I'm showing the precursor +2 ion of this peptide. And then each precursor can be fragmented into the child or transition ions that are shown below here. The fragments are also shown in the chromatogram pane. So each one of these different color traces is a different fragment ion of the peptide that I have selected on the left. You can see the colors correspond to all of the different fragment traces in the chromatogram. You see retention time on the horizontal axis and intensity on the vertical axis. And you see these dashed lines on either side of the chromatogram peak. These are the integration boundaries that Skyline has chosen. You can adjust these in Skyline. We'll be doing this in the hands-on portion of the lecture. And this chromatogram, the quantitative value for this peptide is determined by the integration under the curve of the chromatogram for either left and right boundaries. So I'm going to pause real quick, and we've got a question asking, in theory, the intensity of B and Y ions should have the same intensity for doubly charged ions. But why is it different? The B and Y, so I think the question is asking for a corresponding B and Y ion of the same break, you would maybe expect it to have the same intensity, but that is not the case. So this is a great question, and it really kind of gets at some of the theory of fragmentation in mass spectrometry. You would imagine if this breaks into a B and a Y ion, they should have the same intensity. That's not what we observe. And there are some predictive measures to try to explain why that's the case. I'll point you to some literature because I think it's getting a little advanced for this basics talk, but it's a great question. B and Y ion are not the same. Another question. What is the advantage of monitoring fragment intensities instead of precursor intensities? Fragment intensities are much more specific. So if we have just a single precursor mass to charge, there are a lot of different peptides that might have that same precursor mass to charge. However, not a lot of other peptides will have the same precursor and fragment ion combination, because we know in the mass spectrometer which precursor these fragments are coming from. So it's more specific than using just the precursor. There are ways to use just the precursor. This is called full scan filtering or MS1 filtering. It's just a little noisier. We might see this in DIA later today. Another question. Oh, they're really coming in now. Are you describing the chromatograms from the DIA experiment? I am not describing chromatograms from DIA. Here, we're just kind of focusing on basics of targeted. If not, why the same precursor has been picked for the fragmentation several times. So commonly, you want to pick the same precursor many times for fragmentation because each time you pick that precursor and send it for fragmentation, you get another MS2 scan. MS2 scan being the tandem mass spectrometry scan where you fragmented the precursor and then measured the fragments. So each time you measure the fragments over the elution profile, you get another point across this chromatographic peak. And we will be talking about that more in just a second. Oh my gosh, so many questions now. Why peptides are normally doubly charged? They're not normally. +2s and +3s are most common for bottom-up mass spectrometry proteomics. However, there are a lot of other circumstances where a +2 or a +3 is not most common. So for example, in phosphoproteomics, things like +3 and +4 are more common. Deciding whether you're going to look for a +2 or a +3 or a +4 is part of method development. And commonly, you can use rule of thumb based on amino acids. You can kind of predict whether a +2 or a +3 might be more abundant based on the charges of the amino acids. However, I think commonly in practice, most mass spectrometrists will just shoot the peptide sequence and sorry, my dog's talking in the background. Most mass spectrometrists will shoot the peptide sequence and just see which one is more common, +2, +3, +4. Do you normally consider only a few Y transitions, the top 7, or all the entire Y transitions? So based on what fragmentation you use in your mass spectrometer, B's or Y's or A's and Z's will be more or less common. For PRM method development, you don't have to choose which transitions because your mass spectrometer will measure all of them. And then you can only select, like in the screenshot I have up right now, you can just select the ones that are most intense is a pretty common way to do method development. You'll see in DIA as well, you're measuring all of the fragments. So you don't really have to choose, you just have to decide which ones are the best fragments to measure. What's in the square bracket at the end of the transition in Skyline? So this square bracket in the peptide level is showing you the amino acid position of the first and the last amino acid within the protein. So this is starting at amino acid 36 of ApoA1 and ending at amino acid 46 of ApoA1. The brackets in the child ion tree are showing the rank of the different transitions. So Y6 here is this kind of like light green. It is rank 1, most intense fragment ion, and Y3, this kind of orange guy, is rank 7, the last rank. Ooh, we've got a ton of questions. I will try to get back to them, but I will keep going just for a bit. Hopefully I can answer some of them along the way, though. These are good questions, though. So basic components of a mass spectrometer, I have a little schematic of the guts of a mass spectrometer down here. For those of you in the community that are saying, but Lindsay, you're showing a Q exactive in the ion optics and you're showing a triple quad in the little animated GIF, I know. But I like this animation better, but QEs are very common. So the first step in the components of a mass spectrometer, they all share the same thing. The first step is getting the ionized peptides, the precursors, to enter the instrument. The second step is to separate precursor ions by their mass to charge using what's called a quadrupole. So now we have ionized all of the peptides, they're precursors. Now they're inside the mass spectrometer, and we're selecting for just one precursor mass to charge value. We fragment the precursor. So a population of precursors in the mass spectrometer is bombarded and breaks into all of those different fragments I was showing you in the cartoon before. And then we measure all of the different fragments. Here I am actually showing you the Orbitrap, and we get a mass spectrum. So all the fragments are put together into the measurement for the MS2 fragment spectra. So these are kind of like these four basic components are shared amongst pretty much all of the different mass spectrometers, but the specifics of how the ions are manipulated and guided is a little different based on different types of hardware for a mass spectrometer. Basic steps: ionize the peptides, filter the precursor ions by mass to charge, fragment precursor ions, measure precursors or fragment ions, kind of basic 4 steps. So I'll pause real quick to see if there are some questions I can try to answer quickly. Are the fragments the MS2? Yeah, so this is like, it's kind of confusing, right? Jargon. So the fragments from the precursor are what we measure in an MS2 spectra. MS2 can also be called an MS spectra, it can be called a fragment spectra, it can be called an MS2 spectra. There's a lot of names for it. That's in comparison to a precursor spectra, where you just put all of the precursors in the mass to charge measurement. So you didn't break up the precursors. You can see all of the precursors is in an MS1 spectra. I hope that answered that question. I think we got that one. How many fragment ions are necessary to confirm peptide identification? That's a great way to start a fight at a mass spectrometry conference. Are the most common number of fragment ions I see is between 3 to 5 per precursor, and there's some general rules of thumb that we'll be talking about in a little bit. Typically, you want to pick at 3 to 5 fragment ions that are kind of in the middle of the precursor. So you don't want to just get, like I had showed you that cartoon where it was just like one amino acid and then the rest of the peptide sequence. That's not a very good fragment to choose. But as long as you get 3 to 5 that are pretty much in the middle of your peptide, I think most people would agree that that's good. More is better, and with PRM, you don't really have to choose. How can we quantify peptides with PTM? Great question. You need to make sure that the PTM is represented in the fragment. So that's getting a little ahead of myself. So I'm going to come back to that question later. Having different resolutions for mass spectrometry to scans does not cause important problems for Skyline. Why? So it has to do with how Skyline's processing the data, and there's definitely settings in Skyline to specify the parameters of your scans that will help Skyline process the data and pick the right peaks in your data. So I had in the little workflow, the overview workflow I had shown at the beginning of the slides, I had a reference to a paper we had written a couple of years ago going over the Skyline ecosystem that walks through every step of exactly how Skyline processes the data. If you have co-eluting peptides, can you still identify and quantify them based on MS2? Absolutely, as long as you have distinct precursor or distinct fragment ions to distinguish the two. So if you have co-eluting peptides, but they have a specific fragment ion or a specific precursor mass to charge that distinguishes them, it's totally fine that they're co-eluting. How many fragments depends on signal to noise. Yeah, it does also, how many fragments you pick also depends on signal to noise. We'll be getting into a little bit of that when we go into data curation. Okay, so I'm going to go to these next couple steps. Maybe that'll help answer some more questions too. The required information for doing a parallel reaction monitoring experiment is nice because there's not a ton that you need to know. So you obviously need to know what proteins you're interested in because you need to decide what peptides you want to measure. And then we decide which precursors, which ion charges of those peptides are going to be measured. So for example, let's take this protein, this zinc finger protein. Here we have the entire zinc finger protein sequence, starting with the methionine all the way down to the very last lysine. To build a mass spectrometry proteomics experiment, specifically a parallel reaction monitoring or a PRM experiment, we need to choose 2 to 5 unique peptides from this protein sequence. I've highlighted in blue some different peptides that we might measure by mass spectrometry. In red are all the lysines and arginines, because let's pretend we're going to digest this protein with trypsin. Our job is to pick, of all these different blue amino acid sequences, which ones should we measure? There are a couple ways we can start to narrow down our options. So one, we might want to see which ones have the best mass spectrometry response. So now I've taken each of these peptides in blue. Brian has measured their intensity in the mass spectrometer. Some peptides are very intense and give very high intensity signals in the mass spectrometer. Some don't. You might imagine all of these peptides came from the same protein. So in a perfect world, all of these peptides should be equimolar based on coming from the same protein. So you might also imagine, well, if they're all equimolar peptides, they should all have the same signal. They should all be similarly abundant. But that's clearly, in this figure, we can tell, not the case. Some peptides either digest really well and are released from the protein very easily. Some peptides, so-called, fly very well in the mass spectrometer versus others don't fly very well in the mass spectrometer. Some peptides fragment very well and give very high intensity fragments. Some peptides do not fragment very well and give very low intensity. So I'm telling you all of this, and if you're a biomedical researcher trying to design a mass spectrometry experiment for your pet protein, you might be thinking, well, how am I supposed to know all of these different things? You can source peptides from public repositories. So there's a lot of different resources that you can start from and say, did my protein of interest ever get measured by mass spectrometry? You can ask databases like PRIDE, Peptide Atlas, SRM Atlas, National Institute for Standards. NIST has libraries where they've compiled a whole bunch of different mass spectrometry experiments and said which peptides did give us good signal for different proteins. To show you a couple of these, the McCoss Lab at University of Washington has a website called Passport, where you can look at some different proteins and see the different peptide responses for that protein. So here we have this aminotransferase protein, and clearly the peptide sequence starting with VGG gives extremely good peak area signal. So this is a very good peptide to use if we wanted to measure this particular protein. You can use repositories such as the CPTEK assay portal from National Cancer Institute, NCI. These are very well-defined proteins in this CPTEK portal, where you can see the chromatograms for different peptides. You can even see their analytical figures of merit. So some peptides will be more or less quantitative than others, a little beyond this particular lecture. But in general, if you didn't have your protein in one of these repositories, there's some kind of rules of thumb that can help guide your peptide selection. The most basic rule of thumb is to just ensure that the peptide is actually digested and released from the protein. There's rules of thumb that we'll be kind of going over using Skyline for these different guidelines. If you're doing PTM research, you might want to target specific phosphosites or specific PTM sites that necessitate you have to look at a particular sequence. To show you a little bit what this looks like in Skyline, and then I'll take some more questions. What we can do first is called an in silico digest. So this is where you just take the peptide sequence and, based on whatever enzyme you're going to use to digest, have the computer break up the protein into the different peptides. To do this, you would go to settings, peptide settings, digestion and just set up whatever enzyme you wanted to use for your experiment. And then you can use the filter tab to specify a minimum and a maximum peptide sequence length. So these are some of those rules of thumb where commonly we use minimum peptide lengths because peptides with shorter amino acid sequence are less specific to a given protein. So you might see mass spectrometrists look for a minimum of 8 amino acids for a peptide sequence. You might also specify a maximum length. The longer a peptide is, the harder it is to so-called fly in the mass spectrometer. We'll be doing this hands-on in the second half. You might also use those repositories and use what's called a spectral library based on prior mass spectrometry data. You can say you can narrow down which peptides for a given protein you want to build your assay for. We'll also be doing this in the hands-on section. And just to show you what this looks like, I had mentioned earlier that some peptides, some precursors, fragment better or worse. So on the left and the top, I'm showing you a peptide that has so-called poor fragmentation. So you can see in this MZ range, there's just not a lot of fragment ions being measured for this particular peptide. Versus on the bottom right, I'm showing you a different peptide that has so-called good fragmentation. So you see a full range of fragments across the mass to charge that represent fragmentation all along the amino acid backbone of this particular peptide. We won't talk too much about this, but I wanted to point it out. There's also these transition settings. So for PRM, we don't have to choose our transitions. Our mass spectrometer is going to measure all of the fragments from a precursor. But if you're working on what's called a triple quadrupole and doing selected reaction monitoring, SRM, or multiple reaction monitoring, MRM, you'll have to decide which fragments you want to measure as well. So I just wanted to point this out for those of you that might be doing triple quad work. Okay, so I'm going to pause here and take a couple more questions. How many precursors do we need for good quant info? So you only need one precursor per peptide sequence, but for each protein, you should have at least 2 precursors per protein, at least 2 peptide sequences per protein that you're measuring. I see, what if I am only interested in intracellular part of the receptor that has a limited amount of peptides? I have to compare abundance between different cell lines. So the good news is if you have a protein that is a transmembrane protein, I guess the good news is one of the challenges in mass spectrometry is that sometimes we fail to detect transmembrane domains of proteins. And this has to do with like the hydrophobicity thing I was talking about with liquid chromatography. Transmembrane domains tend to be very hydrophobic anyway, and measuring extremely hydrophobic or extremely hydrophilic peptides by conventional mass spectrometry is tricky. So your intracellular part of a protein is the part that you would probably measure by mass spectrometry anyway. There are products and approaches out there to get more of the membrane-bound proteins, but that's maybe a talk for another day. Is it truly absolute quantification? Is it truly absolute quantification, especially that you're losing 90% of the sample while preparing it? That's another great way to start a fight at a mass spectrometry conference. Does absolute quantification even exist? Some would say yes, some would say no. The point that we're losing 90% of the sample while preparing it, I don't think we really lose that much. And there's a lot of emerging technologies right now that are a lot better at not losing sample. So losing sample is not one of the biggest concerns for the argument of absolute versus relative quantification. Could I use non-tryptic peptides? Absolutely. They might be harder to see because part of the reason everybody loves using trypsin is because it confers a charge to the peptide sequence. But if you have a precursor that you see by mass spectrometry, you can absolutely use a non-tryptic peptide. When one has quantified 5 peptides of the same protein, but 3 are showing differential regulation and 2 are not showing that trend, how do you infer the results in that case? This is a great statistics question. I'm not sure if some of the other presenters will be going over this, but the idea being a protein, all of the peptides of a protein should either all be trending up or all be trending down. And in this scenario, you have some peptides that increase, some peptides that decrease. There are arguments for both camps. Some researchers argue, since we're measuring peptides by mass spectrometry, we're not measuring proteins, all of our differential statistics should be done at the peptide level. There's other camps that argue, but that's not interesting. We want to know the protein level information. So peptide abundances are rolled to protein level. You'll find arguments for both. It's a hard question, and we don't have a good method right now to say exactly if some peptides increase abundance, some peptides decrease abundance, what happened to the protein, but that's an active area of research, I know. How many fragments are necessary to confirm peptide identification? Typically at like 3 to 5. I think 3 to 5 fragment ions is what most people use. Can you be sure? Oh my goodness. How can you be sure your peptide of interest is unique to the protein of interest and not from another protein in the matrix? So one thing you could do is this in silico style approach and take the entire background proteome of your sample and see if that peptide occurs in any other proteins. You could also perform empirical experiments with a peptide standard and spike that peptide standard into multiple different examples of your matrix material to see if the endogenous level of that peptide is changing across different matrices. I think that's good for questions for now. I'm going to go to the next section. So this is just talk a little bit about what I had mentioned before as far as why you would take multiple MS2 measurements for the same precursor multiple times. And this has to do with points across the peak. So I think Brian's probably going to talk about this more for DIA in a bit. But the point is that liquid chromatography has quantitative consequences on your sample. So here I'm showing you a cartoon of a chromatogram. Each one of the vertical lines represents a different MS2 scan, so a different measurement of the same precursor. We measure at the beginning of the elution profile on the left, all the way through the apex of the elution profile, and then finally at the end of the elution profile. We make 1, 2, 3, 4, 5 measurements. We infer the shape of this chromatographic peak, but really all we're measuring is 1, 2, 3, 4, 5 points. We draw this line in afterwards and assume that this blue line describes the ground truth of these specific distinct measurements that we've made. You can imagine if you take less measurements, it's harder to really determine what that underlying blue shape really was. The less measurements you have, the less sure you are of what the shape of that peak is. There's also different consequences of how many measurements you make versus how wide the peak is. So depending on different chromatographic settings, different parameters for your chromatography, you can have shorter fat peaks and you can have tall skinny peaks, and that would affect the shape of the underlying chromatography. The distance between each of these scans in time is called the cycle time. And this becomes very important when you're trying to really build kind of like an advanced method that's measuring a lot of things. So just to point out a couple references that might be helpful, the quantitative accuracy is kind of a function of different parameters of your method. So pointing out on the left here, different settings of a quantitative method can better or worse describe the underlying ground truth of this black chromatographic peak shape. The more measurements you have, for example, this green line with all the points better approximates the ground truth. The less measurements you have in this pink line, it becomes very difficult to measure the underlying ground truth, to put together the underlying ground truth. Brian and I have worked out kind of the sweet spot for a lot of mass spectrometry experiments is somewhere between 6 and 8, ideally closer to 10 measurements across a chromatographic peak. And these are experimental parameters that you can optimize in order to make sure that you're getting the best description of your chromatographic peak. So some of these parameters that you can experiment with are the target list, the scan settings, so the mass spectrometer settings, and the total acquisition time. So the target list refers in parallel reaction monitoring to the number of precursors that you want to measure. If you were doing a SRM or MRM on a triple quad, that would be the number of transitions. For DIA, like Brian's going to talk about next, this is the total number of isolation windows that you're going to use to tile across the precursor range. The acquisition time is the total duration of the acquisition. Commonly in academic mass spectrometry experiments, this is like a 90-minute experiment, but for clinical mass spectrometry proteomics, this is on the scale of like less than 10 minutes. And then finally, some of the scan settings that need to be optimized are dwell time for SRM or MRM experiments, and then settings that control the flow of ions, such as automatic gain control and maximum or ion inject time for PRM and DIA. The target list and the scan settings combined determine the cycle time. And remember, the cycle time is how fast you can sample, you can make MS2 measurements across a peak. So just to give a little bit of background on chromatography details before I take the next question break, the main idea with a chromatography gradient is that you have a separating section of your chromatography gradient. This is where all of the peptides you're interested in should be eluting. This is a function, sorry, this is time on the horizontal axis, and then the percent organic, so how much organic is in the liquid phase of the chromatography. So at low percent organic, most peptides will elute up to about maybe 40% for common proteomics experiments. So hydrophilic peptides elute early, hydrophobic peptides elute late when there's more organic in the solvent. Most chromatography should include a period of washing the column, which is very high organic, to get all the crud off your column before your next sample. And then you re-equilibrate the column. The idea here is that you can increase the total time of your gradient to manipulate the chromatography. You can manipulate the percent organic to affect the shape of the peaks and how many peptides are eluting when. And these are parameters that are specific to whatever peptides, whatever proteins you're trying to measure. So before we get to this second half, I'm going to check for some more questions. The questions have been so good so far. Where am I? So going back to fragmentation, does it matter if the 3 to 5 fragments are used for peptide ID? It doesn't matter if they come from the Y series or the B series. It doesn't necessarily matter. I've been showing a lot of Y because a common fragmentation method produces intense Y fragments, but there's other ways you can use a mass spectrometer to fragment precursor ions that would give you different series. So really you just want to pick the most intense fragment, no matter which fragmentation series it came from, the most intense, and ideally kind of in the middle of the precursor. How many proteins, how many proteins could be quantified at the same time during MRM analysis? So if you're using a triple quadrupole, how many proteins could you measure in a single experiment? It totally depends. So you can really push. So for example, in a PRM experiment, if you're really pushing, you can measure maybe like hundreds of precursors. And if you assume 2 precursors for protein, like maybe you could do like 100 proteins in a single experiment. If you're doing that many things, I think a lot of mass spectrometrists at this point, if you're measuring, you want to measure like hundreds of proteins. I think most mass spectrometrists would tell you to consider moving shotgun either to DDA or DIA experiments. Can you play around with ion collection time, or rather stick to parameters suggested by the vendor for corresponding resolution? I would argue you can absolutely play around with ion collection time. A lot of times the vendor suggestions for different resolutions are specific to, for example, DDA, or specific to PRM, or specific to DIA. If you have the instrument time to optimize these type of things, I would argue go for it. If it's an assay that you want to use a lot in the future. If it's kind of one-off experiment, maybe it doesn't make sense to spend the time to optimize. How long would you suggest the wash step be relative to the column size and other related parameters? This is completely a function of the column. So you should wash about 1 bed length worth of column. And you can calculate the bed length based on just the area, the volume of a cylinder, and then the flow rate of your liquid chromatography, and try to shoot for about 1 bed length worth of wash. And I think 2 or 3 bed lengths to re-equilibrate. Why are you re-equilibrating at 50% B? Because that was just an example. You should re-equilibrate at starting conditions, to be clear. How do you optimize the gradient for each experiment? What is the rule of thumb? Can you please share resources if that goes beyond the scope? Yeah, so exact gradient optimization does go beyond the scope, but I would say you optimize the gradient to try to get whatever peptides you're trying to measure to be as separated as possible. You don't want too many peptides overlapping. And I can post some resources in the documents. So I'll get to a couple more questions in a second, but I want to get to the Skyline tutorial because that'll hopefully like help give you a hands-on. Criteria for determining whether a peptide measured is real or not. So here we're looking at chromatograms like you might see them in Skyline. So a couple things that we look for when we're doing data analysis in Skyline is that all of the fragments shown in the chromatogram overlap, coalute. If they don't, they're probably not from the same precursor. We look for peak shape. So this is a slide from Tina Ludwig, and peak shape ideally should be symmetrical. So this is actually an example, I think, from her of bad peak shape, but it's not terrible. This peak shape is not entirely symmetrical. It kind of like has a little bit of a tail going on the end. So that might be poorer peak shape. And then signal intensity. So now we're showing a very intense chromatogram versus a less intense chromatogram. Most likely, the more intense chromatogram would be coming from the precursor, but we're certainly going to see in the next section on DIA that that's not always the case. If you have additional information, things like correlation to a library spectra can help you be more sure that what you detected is real. Retention time can help you determine if a target has been correctly or incorrectly identified. And then finally, if you have standards, heavy isotope labeled standards that you can spike into your sample, the light and the heavy isotope should perfectly coalute in retention time. That can help you decide that a particular peak is correct or not. To show you a couple of examples, here we have an example of 2 peptides that are displaying interference. So the signal from this particular peptide, we see this nice coelution of the different fragments right at 13.7 minutes retention time. And we see that the peak shape looks really good. And all of these fragments look great, except for this Y4 orange fragment ion. There's a lot of background that this ion is being interfered with. So this would be an example of, I believe this peptide is real, this peak is real and corresponds to this peptide. However, this fragment is not good, and I don't want to use this fragment for quantification. This is another example on the right of a peptide where maybe this peak group looks good at 18.2 minutes in the chromatogram, but this Y6 fragment ion is maybe not a great choice for quantification for this precursor because there's this large, almost co-eluting interference. So we'll be doing a couple more data curation hands-on in just a second. So this is to say that Skyline is good when you have a clean peak group to pick. However, real data can be quite messy and complex. So deciding which one of these chromatographic peak groups corresponds to the peptide, the precursor you want to measure, can be difficult. And this is where having prior information, such as the spectral libraries that we're going to use in a second, can help you determine which one of these two intense, good-looking peak groups is the right peak group for this precursor. So this is the dot product. It measures how similar the spectra are to your library spectra. So this is, like 82% similar, but this other peak group is 99% similar. So we would want to pick this one that is more, gives us more prior knowledge that this peak group is correct. And this is to kind of lead you in when we talk about DIA in a bit. Having more prior information is always better because the DIA data is even messier than any of this PRM data. So while we get set up to do Skyline, I am going to take a couple questions. So I'm going to try to multitask this and figure out the best way to do this. So got a question about, do you usually keep the AGC constant and just change the mass injection time to quantify based on the number of candidate peptides? So I'll say that for PRM, these settings are not something that I typically personally play around with. But this is something in DIA method dev becomes very important. So I'm going to try to do this. We're going to try to do this. So it looks like you can see my screen now. I'm going to just open up Skyline. Hopefully you've got this installed on your machines. So when you open up Skyline, we see this first kind of welcome page. We'll open up a blank document. So in the tutorial that I've sent out, kind of our practice scenario is that there are 6 proteins, clinically relevant proteins that we want to measure in plasma. And let's just say your boss has come up to you and said, here's your 6 protein targets, make me a mass spectrometry assay. Where would you start? So I've got 3 different scenarios for places you might start. The first place you might consider is just starting from nothing. So if you wanted to, for example, start from nothing, the first thing you would probably need is the protein sequences. So I get protein sequences from uniprot.org. To use uniprot for our 6 protein sequences that we're interested in, we would go here to the uniprot.org front page, and then this button retrieve/ID mapping, and type in the proteins that we want. Here I'm going to be using the 6 clinically relevant proteins from the tutorial, but if you wanted to go rogue and use, whatever your favorite protein is, feel free. We just type in the sequences, and I'm just typing in the sequences straight from the tutorial. And now we want to select what these are. What I've entered here are gene names. We want to map these to UniProt, and the organism is human. Okay, submit. Okay, so I entered in 6 genes and I got back 46 UniProt IDs. So for most common proteomics workflows, you might want to just take the 6 reviewed protein sequences. However, we can easily imagine an experiment where we're interested in isoforms, and we might want to use peptides that are specific to single amino acid variants. If that's the case, you would maybe deviate from this workflow and download different isoform variants. I downloaded the FASTA uncompressed just because it's 6 protein sequences, it should be pretty small, and then just dragged that to my desktop. I'm done with UniProt. I go back to Skyline, and now I need to set up my Skyline document. So I'm going to go in the Skyline document to Settings, Peptide Settings, Digestion is the first thing we're going to look at. Let's say in this experiment, I'm going completely vanilla experimental design. I'm going to use trypsin. I'll say I'm not interested in any missed cleavages. I want full cleavage. For prediction tab, we're not going to enter anything here. For our filter, let's say a minimum of 7 amino acids, a maximum of 25, and let's set the exclude N-terminal amino acids to 0. So now we want the full sequence. We don't want to toss out any of the N-terminal. For modifications, I'll keep this as just kind of the standard what I was given, carbaminomethyl. For cysteines, just a common modification that happens when we're working up samples for mass spectrometry proteomics. I'll say 1 variable modification, and the quantification tab we're not going to worry about for now. So that was some of the different peptide settings, and those were just kind of very generic settings. For specific experimental design, you might need to play around with those. I'll go to Settings, Transition Settings next. And under the Filter tab, let's say I'm interested in precursor charges 2 and 3. So this is +2s and +3s, ion charges +1s and +2s. And ion types, let's say B's and Y's. Feel free to go rogue and change some of these settings. For example, ion type, you can also have precursor ion types, precursor charges, maybe your 2s, 3s, and 4s. Feel free to deviate. It's just software, so it'll work for whatever you want to do. I'll hit OK and go to, sorry if you hear my dog in the background. I'll go to File, Import, FASTA, and I will navigate to where I just downloaded that FASTA file, and you should see your 6 proteins populate in the target list on the left. You should also see down in the bottom left corner, it says 6 proteins, 326 peptides, 651 precursors, 8,000, almost 8,500 transitions. We can open up each of these targets like I had shown you in the screenshot. We can open up each of the proteins by clicking on the plus sign next to it. We can open up each of the peptide sequences to show the precursors. And remember, we had just in our settings specified +2s and +3s. And then if we open up a precursor, we see a whole bunch of transitions. So now if we were happy with this and we just wanted to shoot our mass spectrometry sample and see what we see, we could go File, Export, Isolation List for PRM, choose our instrument type. For me, all of my labs have been thermo labs. We would hit OK, and I'm going to export this just to my desktop to show you what it looks like. So this has exported a transition list, an isolation list that looks like this, where for each peptide, we have the +2, the +3, and its mass to charge. So this is kind of if you were to start and do a mass spectrometry experiment from absolutely nothing. Luckily, there's a lot of mass spectrometry data now. So we can instead build our mass spectrometry experiments based on prior knowledge. So in this next section of the tutorial, we're going to use a spectral library that will pretend we downloaded from NIST or PRIDE or Massive, somewhere like that. So I've just deleted my targets. I'm going to reset my Skyline settings. So now I'm starting from another blank document. Now we'll go to settings. Settings, peptide settings, and go to that library tab that we had previously ignored. So I'm going to remove the 2 spectral libraries I had in there so it looks like yours. If we've downloaded a spectral library, we go to Edit List, Add. We name our spectral library May Institute Practice Spectral Library, whatever you want to call it. And then under the path, go to Browse and use your file browser to navigate to, hopefully you downloaded these materials, and it should be under the Plasma folder under Plasma Spectral Library. You should see the spectral library that I've built for you so that we can pretend like we just downloaded this from the internet. Select it, hit OK, and now you should see it populate in the Libraries tab. Check the box next to it to select it, hit OK. And now we're going to import that FASTA again. So we're going to go File, Import, FASTA. So now we've populated again those 6 protein targets just like before. But if you look in the bottom right, we still have 6 proteins like we did before, but now we only have 128 peptides, 128 precursors, and 384 transitions. So now based on the spectral library, we have eliminated a lot of candidate targets for these different proteins. And now we've only included proteins, we've only included precursors that have been observed before in this spectral library. So this is an example of where you're bringing in prior information and you're able to quickly eliminate a lot of potential targets that we saw in the in silico. You can go to View, Library Match, and that will bring up the spectrum from the spectral library. So as we go down for some of the different potential candidate targets for these proteins, we can also start eliminating peptides, precursors that did not fragment very well. So like I had mentioned in the lecture portion, we don't need to measure more than like 2 or 3 precursors per protein. So if we see some precursors that have better fragmentation than others, we can eliminate the poor fragmenting precursors from our target list before we've ever even gone to the mass spectrometer with a sample. And certainly some of these proteins, for example, in my document, ApoA2, only had 2 observed precursors in the spectral library with the settings that we've set up. So I can't eliminate either of those two. I need to keep those. But other proteins like ApoB has a lot of potential candidate precursors, and some of them look better than others. So I can eliminate the precursors that look less with poorer fragmentation. So we started with 0 prior knowledge, just the protein sequence. Now we have downloaded some prior knowledge from a repository, made this Spectral Library to look through what other people have seen. Now, finally, let's imagine we have, like the best case scenario, we have either ordered protein standards for these 6 proteins, or we've made expression vectors for each of these 6 proteins and expressed and purified protein standards just from bacteria. I'll say, no, I don't want to save the changes. In the Skyline workshop directory for this workshop, I have a folder plasma standards. Open up that Skyline document. So now we have a document that I've prepared for you, cooking show style, where we took each of the 6 protein standards. We did just the file export isolation list and measured each of the 6 standards by mass spectrometry. Now we're looking through this first set of data and trying to decide which precursors look good and which precursors are not good quantitative proxies for their protein of origin. So as I look down through, for example, ApoA1, I see a lot of good looking chromatograms. So remember, for good looking chromatograms, we're looking for the peak shape is all the fragments are co-eluting. They're all stacked like a rainbow. The peak shape is fairly symmetrical. It doesn't have shoulders or tails off to one side. We're keeping an eye out for interferences. We're looking for a lot of bands in the rainbow. And then finally, we might consider eliminating precursors from these proteins with lots of precursors. We might just kick out the ones that look worse than our best performers. Okay, so we've got tons of questions. What I'm going to do is in these last, 5 to 10 minutes, I'm going to keep my Skyline document up and kind of go through removing precursors that I don't think look as good as others while I answer questions. I will see how good I am at multitasking. Okay. So first I'm going to quickly scan and see if there were questions people had because they were stuck with Skyline. It looks like everyone's doing okay with the Skyline tutorial at least. So I will try to answer as many questions as possible. And if I don't get to your question, feel free to add it to the Google Doc page and I'll answer in line in the Google Doc page. Okay. How can we quantify peptides with PTMs? So this I had a little bit mentioned before, where we would be looking for fragments specific to that PTM site. This is a little bit advanced in the sense of how do you determine if a fragment is specific to the PTM site. Site localizing fragments is the term you would want to search. I've got a question. What background proteome do you use? The whole uniprot KB. Background proteome will depend on the sample matrix that you're looking at. So if you're doing like a cell culture, the background proteome might be, all of the proteome of what your cell culture is. If you're doing, I'm trying to think, like plasma, maybe you would want a more specific background proteome that is just plasma. Most publications that I have seen for PRM experiments are done on Orbitraps, not much on SciX. Is it because of instrument complexity or just because Orbitrap gives better result? I would say there's just a lot of papers on Orbitraps in general because they're popular. That said, I think TOFs are more popular in Europe, I think. So I think it's just kind of an artifact. There's a lot of paper on Orbitraps because a lot of labs, at least in America, have Orbitraps, unfortunately. Phosphopeptides fly both as +2 and +3. Do you choose both? Oop, it moved. Do you choose both for quantification by summing up or choose +2 for quant and +3 for selectivity? I would definitely say for phospho, just the same as kind of a unmodified proteomics experiment, you would pick the most intense one. You could choose both and do the quantification by summing up the signal from both of them. But I would say it's probably not necessary to use both precursor charges for the same peptide sequence. How could I increase the sensitivity of PRM? Optimization. Yep. You can optimize collision energy. You could optimize fill time or resolution. I think separations and just having cleaner samples is the best way to improve that. So you can do enrichments or orthogonal separations like with differential ion mobility. If we select 5 peptides to quantify a big protein, and there's lots of good flyers, is it important to select them from the whole length of the sequence, like the beginning, middle, and end? That's a great question. If you've got a giant protein and a lot of peptides to pick from, and a lot of them fly well, you might want to try to cover the length of the protein. You might see interesting things like the question we had before, where some peptides at the N or the C terminus of the protein might have different quantitative trends that would signify proteolysis or like degradation of that protein. If you have a lot of options, yeah. When developing an MRM assay, could it be assumed the data acquired from a DIA experiment will translate to the MRM experiment? Wow, great question. I don't know if Brian's going to go into more detail on this, but an MRM experiment is a triple quad experiment. So I would not expect DIA data to look quite like triple quadrupole data, just because the instruments are so different in measuring things so differently. However, I would say DIA data is great for developing PRM assays because the instrumentation is very similar. In the case of sensitivity, precision, and accuracy, which one is the best, MRM or PRM? I think the literature would tell you that MRM, SRM, triple quadrupole will be more sensitive. But I think PRM, that said, even if a triple quad gives you better sensitivity, I think for a lot of things, the sensitivity of a PRM is probably good for most things. If you're doing clinical work with patient samples, definitely go chase that sensitivity dragon. But for a lot of biomedical or kind of academic research, I think PRM is perfectly sensitive enough for most of the things we would do. Does it make sense to survey for an interesting peptide considering different precursor chargers and or potential PTMs? Is it better to survey for very specific precursors? If there's if what you mean by interesting peptide is that there's a lot of PTM sites on that peptide, you might want to consider different precursor charges for that peptide sequence. But to be clear, when you're choosing a precursor to look for example, if this peptide sequence that I have here now, if we imagine the serine at position 4 could be phosphorylated and I don't know, some other strange modification can happen on this peptide as well. That would probably change the precursor mass to charge. unless it was another phosphorylation on the threonine. So if it's an isoform, then the precursor mass to charge that you program into the mass spectrometer will be the same. And you need those site localizing fragments to determine where the modification is. However, if you're looking at, like, say, a histone tail, where you can have an acetylation or a phosphorylation, that'll be a different mass shift. Even if it's the same unmodified peptide sequence, it would be a different mass shift. So you would look for different precursor mass to charge, not different charge states of the same mass, if that makes sense. I think I've got just one more minute left. So if I didn't get to your question, there was a lot of really good ones. Try to put them in the Skyline workshop Google Doc, and I'll get to them there. I'll try to pick a recent one. Can Skyline make precursor selection automatically, like top 5 peptides? You can go to refine, advanced, and use some of these tabs in the advanced refinement. So you could do some refinements here based on peak ranks and pick the top ranked things instead of doing it manually. That said, Skyline can't tell you, like, it will just be intensity. So if there were things like, I don't know, like peak shape, like maybe this particular peptide has a poor peak shape, so I would remove these guys because they got shoulders. I think that's all the time I have for questions now. I'll let you guys take a break before Brian gets on to talk to you more about DIA. Thanks for the questions. Oh, Olga, I think you're muted.
Oh, I am muted. So I was saying thank you, and you have a big, big virtual applause for the entire on behalf of all of us.